Good afternoon. Thanks for the opportunity to speak. My name is Zach Dianis, and I'm one of the urology residents at Duke University, working in collaboration with Dr. Pei Zong, Dr. Glenn Preminger, and Dr. Michael Lipkin as part of the P20 Exploratory Center. Today, I'll present our recent multidisciplinary work, advances in vitro laser lithotripsy research, the role of cavitation, and the development of a novel kidney phantom model. I have no personal disclosures. However, ureteroscopes and laser fibers were donated by Dornier, Boston Scientific, and Luminous. Today's talk will have three parts. First, I'll present our research regarding the role of cavitation in laser lithotripsy. Second, I'll discuss a novel kidney phantom model we developed for laser lithotripsy research. And third, I'll share some preliminary data using this new model to evaluate popcorning laser lithotripsy efficiency and thermal safety. Traditionally, the majority of damage in laser lithotripsy has been attributed to photothermal ablation and microexplosion. It's well known that the laser creates cavitation bubbles at the fiber tip. However, recent work at our lab suggests that the water jet from cavitation bubble collapse could be a substantial contributor to stone damage in dusting laser lithotripsy. Dusting is using the laser to break a larger stone into sub-millimeter fragments for spontaneous passage without the need for basket retrieval. To investigate the role of cavitation bubble collapse in dusting, we designed experiments to isolate the damage of cavitation and photothermal ablation. First, we used a laser fiber perpendicular to a bagostone phantom to compare dusting in water with both a thermal ablation and cavitation to dusting in air, which isolated ablation alone. We conducted experiments at several standoff distances of laser fiber to stone and offset distances from fiber to scope when we used a ureteroscope. Second, we sought to isolate cavitation damage by performing laser lithotripsy with the laser fiber parallel to the stone phantom, eliminating contribution from thermal ablation. In both cases, stone damage was measured using optical commuted tomography. This figure shows stone damage measured by crater volume after perpendicular laser lithotripsy in both water and air. Crater volume was much greater in water compared to air at all standoff distances, suggesting an important role for cavitation and damage. Additionally, in water, damage was greatest at a standoff distance of 0.5 millimeter in pink versus standoff distances of 0 or 1 millimeter, possibly suggesting 0.5 millimeter is the optimal standoff distance for dusting. Next, we isolated damage from thermal ablation while performing laser lithotripsy in water. After observing that at short laser fiber offset distances, bubble collapse had a boundary effect and was distracted away from the stone surface and toward the ureteroscope, we used the ureteroscope to isolate thermal ablation damage. Comparing crater damage of stones treated with and without a ureteroscope, damage was greater without a scope. Evaluating crater volume at several standoff distances, there was significantly greater stone damage without a scope in black compared to in water with a scope in the blue line. Moreover, as offset distance increased, allowing greater bubble collapse toward the stone, crater volume also increased. These findings all support the vital role of cavitation in laser lithotripsy. Last, we evaluated stone damage using the parallel fiber orientation. Though the fiber was parallel, lithotripsy still produced stone damage at several standoff distances. Compared to the perpendicular laser fiber, the parallel orientation still produced approximately 60% of the damage produced by the perpendicular fiber at standoff distances of 0.5 millimeter, again suggesting a prominent role of cavitation in lithotripsy damage. So in conclusion, dusting laser lithotripsy in water produced 2.5 to 90-fold greater stone damage compared to treatment in air across various standoff distances. We propose that this is due to the vital role of cavitation bubble collapse, which was confirmed first using the ureteroscope to mitigate cavitation bubble collapse toward the stone, and then second, using a parallel fiber orientation to isolate cavitation damage. Last, the sweet spot for dusting appears to be a standoff distance of about 0.5 millimeter from the laser fiber tip to the stone surface, which also maximized the damage produced by cavitation bubble collapse. I now want to shift to our second topic, in vitro models for studying laser lithotripsy. While these models are quite advanced in the evaluation of dusting lithotripsy, models to evaluate popcorning laser lithotripsy have been more simplistic. Popcorning is performed with a stationary laser fiber placed into a group of smaller stone fragments, using laser energy to cause fragments to move wildly like popcorn, randomly contacting the laser tip, inducing damage. Previous in vitro test tube models are non-anatomic and have sacrificed renal tissue properties. Therefore, our lab has worked to develop an anatomically accurate kidney model for laser lithotripsy. We started using DICOM images from CT urograms. These were then segmented, and the pelvic caloceal system was simplified slightly to produce our final model. We used 3D printing technology to produce our model mold, and then chose a humimic gel based on relatively similar mechanical, acoustic, and thermal properties to renal tissue. Our final model provides an anatomically accurate in vitro environment to study laser lithotripsy, especially popcorning. Additionally, the translucent construction allows high-speed imaging for continued study of the role of cavitation in laser lithotripsy. This brings us to the third section of the talk, using our new model to evaluate popcorning laser lithotripsy. As industry develops increasingly powerful holmium lasers and the new thulium fiber laser becomes more prevalent, both of which generate significant heat while firing, we're faced with an important question as urologists. What laser and irrigation parameters produce the most effective popcorning, accounting for thermal safety? It's well known that thermal dose determines renal injury based on work by Saporetto and Dewey. Using this formula for thermal dose, which estimates equivalent time at a temperature of 43 degrees Celsius called T43, prior research has estimated a very conservative threshold for irreversible kidney damage at a T43 of 70 minutes. Or in other words, irreversible damage occurs when tissue is exposed to a temperature of 43 degrees Celsius for 70 minutes. As shown by this table, thermal dose accumulates exponentially with temperature increasing our care and not exceeding high temperatures. Therefore, our experimental plan was twofold. First, determine the time to reach thermal dose threshold while firing lasers at various laser and irrigation parameters. And then second, evaluate lithotripsy efficiency, incorporating thermal dose restrictions to lasering time. And we'll focus on the second part today. To evaluate efficiency of lithotripsy, we placed 0.5 gram of stone phantom of known fragment distribution in the upper pole of our kidney model. Incorporating thermal dose data from prior experiments using our kidney phantom, we developed iterative lasing and cooling protocols at each laser parameter for a total time of 2 minutes, limiting thermal dose accumulation while lasering to below a threshold dose of T43 of 70 minutes. Lasering at each parameter was performed with both no flow and a low flow irrigation. After lithotripsy, the stone fragments were sieved, dried, and masked. Laser parameters were chosen based on prior in vitro popcorning efficiency studies and clinical experience. Laser time and cooling time were derived from our thermal dose experiments in the kidney phantom, not presented here. As an example, here's the protocol for 0.4 joules and 110 hertz. Importantly, the laser time for each iteration was shorter with no flow than with low flow, since irrigation sharply attenuates thermal dose accumulation. This is our experimental setup with a ureteroscope tower, a smart irrigation system, fluid tank, Moses 2.0 holmium laser, and a single-use ureteroscope. Zooming in, you can see thermocouples placed at several locations to monitor temperature and thermal dose during lithotripsy. Here are some results. The vertical axis is mass decrease after lithotripsy, which was one of our measures of efficiency. Dark bars are lasering with no flow, and light bars with low flow. First, at higher power settings, efficiency was better with no flow. At lower power settings, this relationship was less clear. Second, the overall most efficient setting was 0.5 joules and 90 hertz with no flow. And of all laser parameters tested at low flow, 0.4 joules and 110 hertz was most efficient. Here, total laser power delivered in watts is plotted versus mass decrease, with circles showing trials with no flow and triangles showing trials with low flow. Again, 0.5 joules and 90 hertz, no flow is labeled as the most efficient setting. This plot clearly demonstrates two findings. First, as expected, as total laser power delivered increases, there's greater stone damage. But second, popcorning at no flow produced more damage compared to low flow, despite lower total laser power delivered. This suggests that popcorn is more efficient without flow, despite laser time restrictions dictated by thermal dose thresholds. So conclusions, kind of evaluated for the first time in a sophisticated kidney model, we found that thermal dose accumulates rapidly without flow, limiting laser time to less than 3 to 4 seconds before cooling was needed. Nonetheless, the most efficient laser parameter for popcorning was 0.5 joules and 90 hertz without flow, accounting for thermal safety. And last, popcorning without flow was more efficient than with low flow. We suspect this is likely related to decreased contact between the laser tip and stone fragments with irrigation, but altered cavitation bubble collapse may also play a role. Moving forward, we hope to evaluate cavitation dynamics within the kidney model using high-speed imaging. which may provide insight regarding decreased popcorning efficiency with irrigation. And last, with increasing thulium fiber laser lithotripsy, we plan to evaluate thermal safety and efficiency in this even less well understood space. I'd like to acknowledge everyone in the Peizong Lab and the Duke Comprehensive Kidney Stone Center, funded by the Exploratory Center. Here's some references, and I'll turn it back to the moderators.